Hi guys, we're going to go over the main points of emphasis for Chapter 7, membrane structure and function. So our concepts to understand and apply are what makes cell membranes permeable, the fluid mosaic model of cell membranes, the different types of molecules and proteins that may be present in the cell membrane, the role of cholesterol in cell membranes and how that affects cell membrane at different temperatures, the types of fatty acids present in the membranes of organisms that live at different temperatures, the roles of proteins in cell membranes, and the role of concentration gradients in the movement of molecules across a membrane. There are a lot of concepts to understand and apply, so we actually have a second slide on this. I want you to be able to describe the types of transport across a membrane and their characteristics, explain passive versus active versus versus bulk transport. How is phagocytosis used by white blood cells? What happens if one of the membrane proteins is mutated, and where might the mutation develop? Explain membrane potential and electrochemical gradient. Describe what osmosis is, how you identify the type of solution a cell is in, whether water or some other particle will move across the membrane in the direction of its movement. The consequences for cells of being from each type of solution, hypotonic, isotonic, or hypertonic, and then also be able to predict the movement of water or other molecules through a semi-permeable membrane. I know that's a lot of concepts to learn, but I think we can review them here, and then you can take some notes again from your book. But let's start here with how does the plasma membrane regulate inbound and outbound traffic? So we have passive transport, active transport, and bulk transport. If molecules are small enough and non-polar enough, they are going to move via passive transport. And this means they don't have an input of energy, but they might require transport proteins. And they're going to move via passive transport down their concentration gradient. Active transport is going to move small molecules against their concentration gradient. They usually require a transport protein, and you're going to have some input of energy in the form of ATP or something else. Larger molecules, on the other hand, move in and out via process of exocytosis or endocytosis. So this figure here illustrates those three mechanisms. Seen in red, passive transport, active transport, and bulk transport. So passive transport basically happens if small molecules don't require energy input, but they're still going to move down their concentration gradient. And you can see that sometimes they move directly across the plasma membrane, and other times they have a transport protein. Active transport requires the input of energy. Sometimes that's from ATP, other times it's from the movement of a different molecule. And then finally, bulk transport is what's required for moving much larger molecules. Exocytosis means large molecules are secreted when a vesicle fuses with the plasma membrane and releases its substances. And endocytosis means large molecules are taken in when the vesicle is formed by pinching of the plasma membrane inward. So cell membranes are fluid mosaics of mainly lipids and proteins, but carbohydrates are also important. The major lipid that's present in cell membranes is phospholipids, and they're unique because they are amphipathic, meaning they have a hydrophobic region that's water-fearing and a hydrophilic region that's water-loving. So if you look off to the right here, you see the hydrophilic region, which is a phospholipid, and that's what's facing water on the outside. And then the hydrophobic region, which are the lipid tails, and they're facing each other on the inside. We use this term fluid mosaic, and that's not a term that everyone is familiar with it, but basically if you look on the right, that's what a mosaic is. And a mosaic is just a picture designed with a bunch of different blocks of different colors. So the idea here is that the cell membrane is a collage of different proteins that are often clustered in groups, and they're embedded in a fluid matrix of the lipid bilayer. And a lot of times when you draw the cell membrane, you just kind of draw it as a line. It's not super exciting, or a lipid bilayer, but really there's a lot to it. You have an extracellular matrix on the outside, and then you have a cytoskeleton on the inside, and you have a bunch of lipids or phospholipids with proteins that are all mingled in. So this is a pretty good representation of what a cell membrane actually looks like. When considering the components of cell membranes, I want you to think about the fats that are in the lipid bilayer, whether they're unsaturated or saturated, and how those will affect things differently. What is the role of cholesterol in cell membranes? And then also proteins. We're going to have peripheral proteins, and those are going to be bound to the surface of the membrane, but they're not going to actually be within the membrane, versus integral proteins. And those ones go through the membrane, so they're actually going to penetrate the hydrophobic core of the membrane. And I'm going to remind you about what we've talked about in class about saturated versus unsaturated fatty acids. Again, we see long chains of hydrocarbons, and with saturated fatty acids, they're all single bonds. But with unsaturated fatty acids, they're going to have some double bonds. As temperatures cools, membranes are going to switch from a fluid state to a more solid state. And the temperature at which a membrane solidifies depends on the types of lipids that are present in those fatty acids. So membranes that have a lot of unsaturated fatty acids are going to be more fluid at cooler temperatures than those that are rich in saturated fatty acids. And in order for membranes to work properly, we need to be able to maintain their fluidity. Cholesterol is another component that's essential in helping to maintain membrane fluidity, specifically in animal cells. So it has different effects at different temperatures. At warm temperatures, such as 37 degrees Celsius, which is about body temperature, it actually makes sure the phospholipids don't move as much. But then as temperature cools, it maintains fluidity, so it prevents them from packing together too tightly. So the idea here is that it keeps the membrane fluid whether the temperature is high or low. Plants have something that's very similar, but it's a different type of steroid, but it's used in a similar way to buffer membrane fluidity. Okay, and as a reminder, we have two major types of membrane proteins. We have peripheral proteins that are bound to the surface of the membrane, but they're not actually in the membrane, and integral proteins that penetrate the hydrophobic core, meaning they go all the way through or partially through the membrane. Transmembrane proteins are proteins that span the membrane all the way through from one side to the other. So we have some examples here of transmembrane proteins and what they can do. They are involved in transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and sometimes attaching the cytoskeleton to the extracellular matrix. So the endomembrane system is super critical in helping us figure out how membrane proteins get to their intended locations. Because if you recall from chapter 6, proteins are made on ribosomes. And ribosomes are either contained in the cytosol or in the rough endoplasmic reticulum. But if we're trying to get proteins to the plasma membrane, which is not in the cytosol or the rough endoplasmic reticulum, we have to get them there somehow. And the endomembrane system is what does this. It transports proteins from the ER, or the endoplasmic reticulum, through to the Golgi apparatus, through vesicles, ultimately so they can either be secreted or placed on the membrane itself. What happens with this fluid mosaic model is that it becomes selectively permeable. The membrane allows passage of some substances more easily than others. This explains how membranes can regulate molecular traffic across the membrane. So let's watch this video.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
A concentration gradient is the difference in the number of molecules or ions of a given substance in two neighboring regions. So when you think about a concentration gradient, you want to think about it as a slide. And molecules move from an area of high concentration down their concentration gradient to an area of low concentration. So when you think about how molecules move across a membrane, you're going to have passive transport where molecules move down their concentration gradient, meaning they're going to move down that slide or slide down that slide. So that is not going to require the input of energy. Instead, they're just going to slide down the slide without the input of energy via a process called diffusion, or sometimes facilitated diffusion if there's a protein channel or something else there that's required for them to cross the membrane. Active transport, on the other hand, those molecules move up their concentration gradient. So think of this as if they're walking up the slide backwards from an area of low concentration to an area of high concentration. In that case, they're going to require the input of energy. That's going to come from ATP or from the movement of another ion. So in the example they showed you in the video, instead of ATP, glucose was brought up the concentration gradient by moving sodium down its concentration gradient. Passive transport is the diffusion of a substance across a membrane with no energy investment. Diffusion means there's a movement of particles of any substance, so they spread out evenly in the available space. So if you look at the top set of images and the orange molecules of dye, they start really concentrated on the left and they have a net diffusion to the right because they cross the membrane until they reach equilibrium. However, if you look at the ones on the bottom, you see a bunch of the orange molecules on the left and a bunch of the purple molecules on the right. And the net diffusion is in opposite directions until they reach what we call dynamic equilibrium. And it doesn't mean they actually stop moving, even though the concentrations are the same. It's just that as many molecules cross the membrane in one direction and the other. That's why the arrows go in both directions. So even though the molecules move randomly, diffusion of a molecule can be directional in order to even out the concentration on either side of that membrane. So if you think about that concentration gradient and that idea of a slide again, the steeper the concentration gradient, the faster the diffusion will take place. So with a really steep concentration gradient, you're going to have a really fast rate of diffusion. Whereas with a low concentration gradient, diffusion is going to take a lot longer and be a lot slower. Let's review this idea of facilitated diffusion. Really, it's still passive transport, but it's enabled by proteins that are within the membrane. In facilitated diffusion, transport proteins speed the passive movement of molecules across the plasma membrane because they allow for movement of some molecules that might not make it through. Transport proteins can include channel proteins like the one on the top, or carrier proteins that open on one side and then by binding to that solute, they open to the other side. So here's an example in real life of what can go wrong if you don't have a transport protein that's necessary for diffusion. So people with cystic fibrosis have a mutation in the CFTR transmembrane protein. CFTR stands for cystic fibrosis transmembrane conductance regulator. And basically, this is a channel that allows chloride ions to cross the plasma membrane. And sometimes people will have no protein at all, or sometimes they have less protein, depending on the different types of CFTR mutations they might have. So cystic fibrosis is a genetic disease and it affects about 30,000 people in the United States. Some of the symptoms include a buildup of mucus in the lungs, and that's because you don't have chloride ions that are excreted in order to make the mucus thinner. It's also building up of mucus in the pancreas and other organs. It causes frequent coughing and sometimes producing phlegm and blood. Because of that, individuals have respiratory infections and difficulty breathing, and they also tend to have difficulty digesting and absorbing nutrients from food, and that leads to poor growth and low weight gain. They have salty skin, difficult bowel movements, and infertility. And I have this really cool YouTube video that I was going to show in class, but I'm just going to post it on Populi so you can check out how that transporter allows chloride ions to work effectively or be excreted effectively. Active transport uses energy to move solutes against their concentration gradient or up their concentration gradient, meaning you're basically walking up the slide backwards, and obviously you're going to need an input of energy. They're showing you an example here where sodium is pushed out of the cell from an area of low concentration to a high concentration, and then potassium is brought into the cell up its concentration gradient as well. So in order to do this, you need ATP to provide energy for this transporter. It's a sodium potassium antiporter here. So we have this unique example of what happens in most cells. Most cells have a membrane potential. And what that means is that there's a voltage difference across the membrane because there's a different distribution of cations and anions. So let's remind ourselves what those terms mean. Cations and anions. Cations, positively charged molecules, anions, negatively charged molecules. So those two combined forces work together to drive what we call the electrochemical gradient. And this is going to drive diffusion of ions across the membrane. A chemical force is the ion's concentration gradient itself. So here we have hydrogen ion being pumped out of the cell to a greater concentration gradient, right? And then an electrical force, the effect of the membrane potential on the ion's movement. As you can see, it's being pumped out of the cell, so positively charged hydrogen ions are being pumped out, and it makes the membrane itself, or the area around the membrane on the outside, a lot more positively charged. So if you look at the two of them, we have an electrochemical gradient. We have a difference in the chemicals on either side, and we have a difference in the charge on either side of the membrane. And as you can see, hydrogen ions being pumped out, and we have to use ATP in order to drive this proton pump because it's going against its electrical gradient and chemical gradient. And what's great about this electrochemical gradient is that it can be used to drive other types of transport in the cell. So in this example, we show how the proton pump is making hydrogen ion be really concentrated on the outside of the cell. So naturally, hydrogen ion is going to want to diffuse in. So what we have is a transporter that couples transport of hydrogen ion going down its concentration gradient with the transport of sucrose going up its concentration gradient. So we don't see the use of ATP in this transporter example. We see the hydrogen ion energy being used to transport the sucrose, right? So this is called co-transport, and it occurs when active transport of a solute indirectly drives transport of other substances. So we're not using ATP, it's indirectly driven by other substances going down their concentration gradient. The downhill diffusion of the solute, so in this case this is the hydrogen ion, is coupled to the uphill transport of a second substance, in this case sucrose, against its own concentration gradient. Small molecules in water can enter or leave the cell through the lipid bilayer or via transport proteins. However, larger molecules such as polysaccharides and proteins cross the membrane in bulk inside vesicles. Bulk transport across the plasma membrane is going to occur via exocytosis or endocytosis. Here we have a few examples of endocytosis. Phagocytosis is where the cell itself goes and grabs a food particle or something else and pulls it in. Pinocytosis is where you create a tiny little coated vesicle and pull those solutes in. And you can also have receptor-mediated endocytosis, where particular receptors are present on the outside of the cell, and so only those specific solutes bound to the receptors are going to come into the cell. We should point out that osmosis is basically a type of passive transport of water. It's like diffusion of water, but we call it its own thing because it is the solvent, not the solute. So water molecules are going to move across the selectively permeable membrane, and they move from a region of low solute concentration to a region of high solute concentration. And the reason they do that is to dilute the high solute concentration. So water keeps moving until the solute concentration on either side of the selectively permeable membrane is equal. You need to be able to define these terms when you're talking about tonicity of a solution. So an isotonic solution means the solute concentration is the same as that inside the cell. So that means there'll be no net water movement across the plasma membrane. It's going to stay the same. Hypertonic means the solute concentration is greater in the solution than inside the cell. So that means the cell will lose water because water will be moving via osmosis out of the cell. A hypotonic solution means that the solute concentration is less than that inside the cell. That would mean that the cell would gain water if it's placed inside a hypotonic solution. Cells without cell walls are going to shrivel in a hypertonic solution and lyse or burst in a hypotonic solution. So animal cells don't have a cell wall, so that means that they're going to lyse in a hypotonic solution. They're going to remain normal in an isotonic solution. They're going to shrivel up in a hypertonic solution. Whereas plant cells have a cell wall. And actually, if you think about it, when you water them with water. So they're going to be turgid when you water them with water because they're going to be placed in a hypotonic solution. Isotonic means that they're going to be flaccid because the net flow of water in and out is going to be the same. And if you place them in a hypertonic solution, you're going to have what's called plasmolysis. And I actually have a video that I'm going to post on Populi via this link that you can look at to understand what plasmolysis is. So you already did the case studies, and that should make all of this make more sense. And finally, as a reminder, you need to finish up group activity number 3. That is due on Monday. You need to describe the 10 organelles and the 7 types of transport due Monday, and then peer reviews are due on Friday.